Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Guido Van Marle. He's an associate professor at University of Calgary in the Department of Microbiology, Immunology, and Infectious Diseases. So we're going to talk about some of his research surrounding HIV. So Guido, thanks for coming.
Thank you. Thanks for inviting me.
Yeah, tell me about your HIV research. What are you looking at?
Okay, so in HIV, so I'm trained as a virologist, and over the years of my training and fellowships, I became very interested in viral evolution and looking at how HIV evolves in HIV-infected individuals. So that is sort of the mainstream of my research at the moment, is where we look at how does the virus evolve in a particular infected individual, how do they differ between individuals, and sort of figuring out where the virus hangs out basically in a infected individual, and more or less also where do they hide out, because there's a lot of debate on, or a lot of interest in what we call the reservoirs. So a patient gets infected with HIV, we know we have no vaccines, there's no real cure, although we had the bone marrow transplant, and the example of that was Timothy Brown, who recently passed away from leukemia, or from cancer, not necessarily from HIV, that got cured by the bone marrow transplant. But that is
how many years was he cured of AIDS?
Well, when did that happen? That was a couple of years ago. So he was not on therapy. He called himself the unluckiest person, became the luckiest person, because he had HIV and then he developed a leukemia. And so they had to give him a bone marrow transplant. And they actually gave him a bone marrow transplant with cells that are resistant to particular types of HIV infection, because they miss a receptor or a molecule on the surface called CCR5. And he got a bone marrow transplant from an individual that what they call delta 32 CCR5, which is a polymorphism that makes this receptor non-functional. It's not present on the surface of the cell. So the virus can't use it to infect these cells. So basically, he got infected, or he got transplanted with cells, with an immune cells that were now resistant to HIV infection. And he was basically functionally cured. Probably there was some virus hiding out in other areas of the body, because not every cell was replaced, only cells of the immune system, but he could live without therapy. And there are some other patients that have been treated with that successfully ever since. But that is the only real example of a potentially cured individual. There might still be virus hanging out in some deep reservoirs, which is one of the things we're interested in. But compared to somebody else, yes, he was functionally cured. So anyway.
Well, because some people have latent HIV infections that don't become full-blown problems for them.
Yeah. So we have what we call long-term nonprogressions. Those are people that are infected with HIV, but they don't develop what we call HIV/AIDS, so the acquired immunodeficiency syndrome. But they don't develop it, but they also don't need treatment. So they're not treated, and so they keep, for some reason, and those individuals that we are very interested in, to figure out how that works. How do they keep their virus at bay? How do they prevent themselves or to progress towards this immunodeficiency that in the end will kill you? Because the virus itself, despite some direct pathology that it causes, its main pathology is destroying the immune systems so that you become susceptible to super infections and you die from those. So there are some
let's get into some of the mechanisms. How does it hurt the immune system in people? What happens?
Okay, so that's also one. done a lot of progress also with regard to therapy. So now we can keep people alive with these combination antiretroviral therapies. We're probably going to go into those later. So what happens? So HIV infects immune cells. And we'll talk about what we call CD4 positive T cells. So those, and there are some other CD4 positive cells like macrophages and other cells that can be infected. Even neurons can be infected, but they are not producing any virus. There are some other cells in the brain, such as astrocytes, that can be infected, but it's not really productive or happy or a big infection, but they still produce some virus. And there's microglia, which are macrophages that hang out in the brain. So macrophages are cells of the immune system that we float around in the blood too, that can be infected too. So it's not just T cells. So T cells are these, these people know maybe as T helper cells, and there's a whole broad range of them. But those immune cells and the macrophages are infected by HIV. And so when in an infection or in the acute infection, when somebody gets infected, the virus replicates very highly. It destroys some part of the immune system. We're going to go into that later. You get a dip in the CD4 T cells or the immune cells, but you don't get that overt immune deficiency. And then sort of the virus replication dies down or becomes self-limited. The immune system comes back up. And then you get in sort of like so in that phase, during that acute infection, people present themselves with like flu-like symptoms. And so they feel crappy, they have feverish, but nobody, will directly say, well, I've got a cold. Right. Now probably will say, maybe I have SARS coronavirus too, or COVID, but nothing that would say, oh, yeah, I'm infected with HIV. And then their immune system recovers, and the virus basically goes in sort of a dormant state. It's not really a latent state. And latency is where we think about, for instance, like herpes viruses, where the cells are infected and then the virus just hangs around as an extra piece of DNA in the cell, doesn't do anything. HIV, there is a mixture. So in HIV, in that asymptomatic phase, there are cells that have virus in them and don't produce any viral progeny. They don't produce any virus. And there are some cells that produce some virus. But probably the virus that's being made is being controlled by some of the immune responses, and so by some immunity. And that's where when we come into long-term non-progressors, so how do you keep controlling that? And then over time, the virus stays there, but it still induces inflammation. There's still the immune system is responding, and some of the immune cells are dying because of the HIV and of the inflammation. And then the bone marrow can still, where the immune cells come from, can still keep up with that and keep making new and replace those immune cells that are dying. But then at a certain time point, the immune system, or basically the bone marrow, wears out in a sense, or gets tired, can't keep up anymore. And the immune system starts to, so you don't replace those cells that are dying, so more cells are dying than are being replaced. And now you see your immune system, your CD4 T cells in the periphery. I'm going to go a little bit later. So,
well, look, if you look at the blood of someone who's a long-term non-progressor versus someone that's actively sick, what are their CD4 cells look like? How are they different? What do their other immune cells look like?
So what you will see in somebody who's immune compromised, the CD4 cells are low, below 200, and you see a lot of virus. In a long-term non-progressor, we see normal CD4, maybe a little bit lower CD4 T cells, and we don't see actively replicating virus or very low levels. Both individuals will have HIV in their cells. HIV is a retrovirus, so what it does, once it gets into the cells, its genome, that's normally an RNA molecule, that's a different type of genetic material than the DNA that we have in our cells. It copies itself into a DNA and then inserts its genome into our genome. So it will become part of our chromosomes. And if it's silent like that, it can stay as long in the cell as long as that cell is alive. If that cell gets activated, we can wake up the virus and the virus comes out. Once the virus is replicating, then it induces all these inflammatory responses or these immune responses that then wears out the immune, that then induces other immune cells that are not infected to die. And so there are cells infected by HIV, and those cells die because of the HIV infection directly. But there are also a lot of what we call bystander cells that are not infected, that are basically turning over and dying because they're activating and their normal immune pattern is disturbed. And that is something that doesn't, in people that are long-term non-progressors, sometimes that inflammatory response is not there. So, or it's really dampened. So there's not a big assault even during that asymptomatic stage on their immune system, as well as they actually also maybe make antibodies, or they have an immune system that can keep up, that recognizes HIV. And even though HIV changes a lot, even if it changes, the immune system knows how to recognize those new varieties and basically keep the virus under control, and therefore under a lower level of infection, and sometimes not even directly detectable with classical assays. You have to find some other things. And in that sense, stay out of that HIV/AIDS stage so that their immune system doesn't die off. And that
have you looked at CD4 cells again in these two kinds of people?
Yeah.
Not only there's more of them obviously in the the non-progressive, but what else is different about the cells themselves? Do they have different metabolomics? I mean, what can you see?
Well, that's where people are looking at from what is the difference between those cells. And some of it is also that those cells, so when you take a look, when the cells is dormant, you might not see any difference. But when they get activated, if you would take somebody who would progress towards disease versus somebody who doesn't progress towards disease, sometimes they're immune, and it differs per individual too. Right? It's not just one mechanism fits all. But some these individuals have either a different virus in them that is less pathogenic. Others have a virus or have the same virus, but their immune system doesn't overreact to it. So that you don't get these massive inflammatory responses that then leads into immune cell killing. Because normally the immune system kills, if you get an immune response to any pathogen, it turns on, produces a lot of cells, and then once it's cleared the infection, all those immune cells that are focused on fighting this one infection are being killed off, because otherwise your blood would overflow or get stuck with all these cells. So it kills off the cells it doesn't need anymore, and you will only leave some memory cells or cells behind that are ready to be activated again when you get a new infection. And so HIV messes up some of those pathways so that you actually cells start to get dying. And so that is also to do with inflammation and immune regulators. And some of those immune regulators, and there are a whole bunch that people have identified, that is different in long-term non-progressors too. So there is immune control, so they recognize the virus and keep it under control, and thereby control the infection and also avoid you from developing AIDS, HIV/AIDS. And then there is the cells are not responding weirdly or inappropriately, so wipe out your immune system. And there's some HIV variants identify who do not induce that strong inflammatory response either. So there's both a host side, and so the infected individual, as well as the virus side.
All right. Well, where normally are immune cells taken out of circulation? Does it happen in the spleen, or does it happen in other parts of the body? Like, which organs are places where these cells are harbored once they've been created?
So you talk about, you mentioned the spleen, and you mentioned the lymph nodes, and so those are some of the classical immune organs. People always say, HIV infects the cells of the immune system. The immune system is all that stuff that goes through our peripheral blood. So basically the stuff that runs through our veins, through our artery veins.
If you like this podcast, Please click the link in the description to subscribe and review us on iTunes.
It's not the only site. And one of the things that we look at together with the people here at the Southern Alberta HIV clinic with Dr. John Gill, that I work very closely, a clinician and a medical director who's been working with HIV patients since the start of the epidemic, that not only the cells of the immune system in those obvious tissues, such as bone marrow or lymph nodes, are infected. Other cells, there's immune cells hanging out everywhere in other tissues too. They're in the brain, but there is also this very important tissue that we call the GALT, or the gut-associated lymphoid tissue. So that is basically all those lymphoid cells, so all those immune cells, and there are different, types of lymph nodes that line our gastrointestinal tract. And there's a lot of immune cells there. And that is, of course, a main target of HIV too, because these cells can be infected by HIV. It is very logical that we have a lot of those immune cells in the gastrointestinal tract because it's actually open. It's basically exposed to the outside. We eat every day, we swallow stuff. So it's basically outside. Even though it may look inside, it actually is outside. So there's all these pathogens, good and bad bacteria, and viruses, good and bad viruses coming in there, and you need to have that line of defense. So what happens is that actually what we figured out, that there's a lot of infection going on, also early in infection in an HIV-infected individual, in those cells in the gastrointestinal tract. And so during primary infection, during that acute infection, a lot of those cells actually die. And so your immune system gets a big hit during early infection in that tissue. And then you see somebody recover.
So that'd be a problem, because then everything you eat would leak into you, and you'd all of a sudden be under assault.
Yeah. So, and that is some of the things where we talk about something like leaky gut. And so that basically also that inflammation causes your cells that normally are nicely covering your gut, causes disruptions in there. So stuff can leak in. And then what we call leaky gut. So some bacterial products can leak into the periphery, into the bloodstream. And then the bloodstream, or we would respond to that and with inflammation. So that would exacerbate inflammation and make things even worse. And so then HIV causes damage, then bad stuff comes in, and that causes also damage. So together they create damage. So there is also some ideas that there is also a gut microbiome issue. So the bacteria in the gut, maybe they're working together to keep that inflammation at bay too, because some of the bacteria in your gut actually help reducing this inflammation, because you don't want your gastrointestinal tract to overreact to everything that's in there, the good things too. So, but it is an issue, but it's also an issue. It is this, all those immune cells go there and then they go there to die. And what people have also observed, that if you give somebody therapy, antiretroviral therapy, and so if somebody during acute infection loses those immune cells in the gut, then I said the immune cell comes back in the blood, but not as much in the periphery or in the gastrointestinal tract. The virus keeps replicating there and keeps chewing away there at that immune system, even though in the blood it looks okay. And so there's these cells that go to the gastrointestinal tract basically to die, and that helps to the immune depletion. And then there is also a thing, I'm going to give somebody retroviral therapy. And so if somebody develops AIDS, or, you give them early on, give them therapy before they even develop HIV/AIDS, and you control viral replication. That in general shows in the periphery, you get a replenishment of the immune system in the blood. You see that, your CD4 cells go up, and maybe not back to what it normally maybe was, but really in a good stage. The immune cells seem to be behaving relatively normal. There's still some of them cases a bit dysfunctional, but what you see in the gastrointestinal tract, that population doesn't restore as quickly. It stays behind. So in a sense, you're still immune compromised in your gut.
Yeah. What can people do for interventions there?
So
I know diet. I would think diet would have a big effect in changing it, and then the microbiome of your gut would probably be altered dramatically.
Yeah. So the gut microbiome is altered dramatically. We don't know how that works, or we're getting close to understanding it. The thing that it is, antiretroviral therapy is still the best approach because getting newer and different drugs that also target the virus in that compartment, in those reservoirs, because not every antiretroviral drug penetrates every tissue as efficiently. But with a lot of other, over the years that we've been working with them, with these patients and with the antiviral therapies, we've come up with some very good approaches to sort of keep that virus at bay. The dream is, of course, to get people off these drugs because it's, of course, not a walk in the park. There's some toxicity associated in it. But compared to what it used in the early days, it's a world of difference. And even compared to the early combination therapy, so you get therapies of multiple different drugs at one time, because if you only would give somebody one drug, then the virus would have an easy time to become drug resistant because it can mutate. But if you give multiple different drugs at the same time, you make it actually very hard for the virus to become resistant to all of them.
Quick question here. What happens if you genetically sequence someone with AIDS? Will the viral DNA show up, and will your sequence be altered?
You will find, or you mean your sequence, your DNA, or you mean the viral DNA?
My DNA, if I get it sequenced, and I have AIDS, will it look different? Yeah. And can you identify specifically where the insertions are,
Okay. So we can figure out which cells or which parts of the genome the virus hangs out in. Yeah? So not every cell is infected. But if we get some of the cells, then we can sort of figure out where did this virus, when it gets inserted, we can figure out by sequencing the genome or the DNA in the cells that are infected, we have some tricks to figure out which parts of your chromosomes does it hang out in which parts. And we see that it's I wouldn't say it's random, and it's not. That there are places that the virus likes to go, or when cells, you have to see when a cell is active, not all the genes in a cell are active. And each cell in your body, so an immune cell versus a skin cell, they have different functions. So certain times they need a gene active and other genes don't need to be active. And what the cells do is what they call chromatin, is these proteins that basically condense or fold up that DNA that is not active, that they don't need the genes to be expressed there, into a compact, sort of like a ball of yarn. Yeah, it's basically like this chromatin is dense, and then the virus cannot integrate it. So it will be integrating in those pieces that are active. Now, cells go through cycles that are active and not active. So if you end up in an area that was first active and then inactive, then you might find it in a different reason in a cell that has one area that is always active. So you could see different areas where the virus hangs out in. And there might be some ways that in certain people, it ends up in these areas that get silenced and they never wake up. Those cells never wake up, never get activated, and the virus never can get out. So that may be different. Or it lands by accident in particular spots that will never be active. And so the virus will never be activated. That is possible. And so, but yes, we see differences between individuals, the spots where it hangs out in. But we haven't, we're not as good yet, or we haven't analyzed enough patients to figure out a pattern there. There are some patterns where we see from, maybe somebody who doesn't progress towards AIDS that long, the virus hangs out in a particular other part than somebody who rapidly progresses. And can we sort of push and trick the virus to go to, or keep it there so that it never wakes up?
What do you mean it hangs out in a different part? What do you mean?
So it hangs out in a different part of your genetic sequence. So in all your DNA that you have, then it maybe hangs out in particular one area where there are particular genes, and in another area where it's previously. You have to see your DNA as a long strand, and it's multiple different strands. So the virus can hang out in the different strands, but also within each strand, it can maybe hang out at the beginning, somewhere in the middle, or maybe at the end. So it can insert its genome in different areas in the human genome. I hope that makes sense.
Huh. Okay, so you've seen in looking at people's DNA that it's, I mean, how many different areas? Is there just a few clusters, or is there a lot?
There are some clusters, and there's quite some different areas too. So in immune cells, there are certain genes that are active, and that's where you see a lot of the virus basically ended up, because the virus or the DNA is available, so it can integrate in there. But then if a cell goes dormant in those areas, then it basically gets compacted, and then you find it in an area that is really compacted. So it depends a bit on the cell cycle, or the state that the cell is in when it gets infected. If it always stays active, then gets sort of like with memory cells, they might get sort of immune memory cells that get sort of shut down. They hang around, they just live, but they're not really active to produce because there's no need for those cells to be activated as part of immune response. So then you find it in those areas that are then actually very transcriptionally, or like the genes expression in those areas is very low. And so in general, if the gene expression is high in an area, virus likes to go there because it's available for the enzymes to hold on to the DNA and stick your DNA in there. So, and that changes by cells. And we're looking at that, but there's also each piece of DNA is slightly different. And so that also means it lands maybe in an area that is very active, and so the virus will be active too.
Okay.
So it's very complicated, or it seems like very complicated.
Has anyone looked at other HERVs, human endogenous retroviruses, and compared them to HIV? Like this is probably our first glance at a HERV that just has entered into someone. So I would think we can learn a lot from it. When it integrates, is it fully functional? Does it cleave off any of the DNA that composes the HIV or any of the original nucleotides?
You mentioned a very interesting point about HERVs. So it's like human endogenous retroviruses. These are these remnants these are basically retroviral sequences. So viruses similar to like HIV, but not HIV, that we probably acquired in ancient history, and maybe some of a more recent history, and they became part of our genomes, and they are passed on to our offspring, and we live with those. So HERVs, and so if you take a look at human endogenous retroviruses, there are HERV sequences, viral sequences in us that are completely functional. They would produce a functional virus, can infect new cells, and that happens while we speak. But they don't seem to cause disease. There are some examples where we now find that maybe they play a role in MS, but the bulk of them don't. There's also a lot of these HERVs deletion mutants. So they have pieces missing, make no infectious virus. If you actually take a look at HIV, when HIV does its replication and integrates the same way as a HERV does, there is a bunch of these HIV genomes and viral variants in our genome, and a significant amount that are actually non-functional. Or they produce virus, but they're missing pieces, or they're not as good, they're not a functional virus. So you can see that there's a property of the virus. It makes inactive viruses too, but it makes so many good viruses. It doesn't, if it has a consciousness like we humans would, we would consider it as a human, it doesn't care because there's still enough offspring. So those endogenous retroviruses, what has happened with those is we learned how to live with those. So they don't no longer cause disease, except in particular situations where MS, where they have been implicated. But, there's still a lot of debate about some of those things, but there are some therapies focusing on that they actually control, or what controls it, why, because we all have those. Why does it go wrong in an MS patient and not in somebody with multiple sclerosis and not with somebody else? But those viruses are part of our genome, and we could have acquired them like thousands of years ago. And they might have gone, maybe they caused an HIV-like illness or some other illnesses. And then maybe what is left is the people, or maybe the long-term non-progressors. And what also happens then, they infect the cells of our germline. So they infect our sperm and our eggs. Those cells get integrated in there and then pass on to our offspring. And HIV doesn't infect the germline cells as of yet. So we will not pass it on like that through sperm to the DNA in our sperm cells and in our eggs, in our oocytes. If a child gets infected, this infection happens after gestation in the uterus. So, but what has happened with those endogenous retroviruses, that is probably, they ended up in our genome, in the sperm cell, and then get passed on. So all those deletants and even intact viruses. And some of those viruses have just as many genes as HIV. They look quite different, but they could potentially be that. And other organisms, other mammals have that too. So you find them in pigs, you find them in monkeys and chimps, you find them in koalas. And there is now some evidence that in koalas, you see that effect of a retrovirus like HIV or their equivalents actually starting to become an endogenous retrovirus and becoming part of their genome. And they might actually utilize this to protect ourselves.
Well, what's happening with HIV? Have there been children born not just sick from HIV, but born with the virus pre-endogenized into their DNA?
Not that I know of. And it's very hard because you need to go to the germ cell lines, and because the cells don't have those receptors. And so the cells are not susceptible to HIV directly, so they have to come in through another route like that. So what happens the child is protected in utero, but at a certain point, it still gets exposed to some of the bloodstream of course, the mother, and that can induce that can set off an infection. So it can. But what we also have seen is that there, and that's how we figured out too. Is like a child from an HIV-infected mother is HIV negative and can be born HIV negative, so not being infected, if we take certain precautions. So if we put the mother on antiviral therapy, so you block the virus or you reduce the virus, and actually deliver the child through like a cesarean, sort of C-section, because then you take the child out with as little blood exposure as possible. Because then during birth, of course, when it comes out of the birth canal, that is quite, an intense experience where there's also a lot of blood, the child gets exposed, and that way it can get infected. What they then also do is they put the child on antiviral therapy, and that would also reduce the chance if they get infected that way.
Well, what's happened to children that are born with no antiviral therapy? Does HIV
and if they have HIV
kill them,
infected,
or does it spare them?
So if they get infected, they will develop HIV/AIDS
at a very early age and still suffer all the problems.
And we had the Mississippi baby where they thought that they hit the child hard, or hit the child, but gave the child with intense antiviral therapy at the start. Then you actually hopefully try to prevent, if the virus got into the child during birth, you try to prevent it from infecting anything, and maybe you can sort of cure it and prevent infection. And in some cases that might work. And in some cases with the Mississippi baby, they thought, so that was from a woman from Mississippi, that's why they called it the Mississippi. This child was on that early therapy, then lost to follow-up, and they thought it was cured. But then the virus was probably hiding, so you kept it under control for a long time, but then it came back anyway, so this child had to be on antiviral therapy. So if you don't give him antivirals, unless if the child is lucky enough to be a long-term non-progressor, invariably, or in the vast majority, you have to keep antiviral therapy to keep the child alive.
So if you compare again, back to gene sequencing, if you look at non-progressors versus people that have full-blown AIDS, have they sequenced the genes of both of those groups to see if there's a difference in where the HIV inserted?
Yeah, so in many cases, this has to do with some of these antiviral responses. So there are these polymorphisms that they have, in particular genes that have to do with antiviral responses and how they respond to those. And that's where the differences lie. But there is also an interesting thing, is like, I wouldn't say that not everybody is the same, but there's a lot of variability in it. So one gene set of genes is protective in a subset of patients of these long-term non-progressive. In another subset, it's another pair of genes, and it's a combination of them. Yeah. And if we look at our natural hosts, which we think HIV came from SIV, simian immunodeficiency virus. So that came from simians, so primates and macaques, so chimps and gorillas, or pseudomingabes. And what people have seen in the natural host, that SIV that's been around also 20,000 years, that those animals to a certain extent have to learn to live with that virus. It's not endogenized, it's exogenous. So it's like HIV, it needs to be transmitted from animal to animal, not from vertical, from mother to child directly in the germ cell lines. Those animals have virus, lots of virus in their periphery, in their blood, but they don't have any disease. And the reason why that is because certain immune pathways are not activated. So they tolerate the virus, so they live with each other. And that's probably what you need to get also to become in the end an endogenized virus. But similar things are observed in humans, but that is, not as common as we think. There are some examples of actually people who are protected from infection, so they get exposed, but then the virus for some reason can't get a foothold.
Well, but if it gets into enough members of a population, when they breed, they would successfully breed with ones where it's the other members are infected but not sick, so it could change the whole. The whole population. Yeah.
So that's what if you would nature take its course, in the end, that would happen. But then if you take a look at that, at the current the cost to the species in certain way, so it could wipe a species out, push a species out in the to the brink of extinction. That the population is too small to survive, but there is also the population that could survive. But on the other hand, too, there are some things, there are long-term non-progressors that do particular things that in theory everybody can do, or the majority of the people can do, but for some reason the immune system does not decide to do that. And so there is some training or thing going on that, we have those defense mechanisms in us, but some individuals do that right from the start and keep doing that, and others do that for a while and then lose that propensity over time, and some people that never do it. Yeah, so it is, this is the beauty of nature. That's also why nature is so plentiful on our planet. This is in a sense, this natural process where there's this dance between the virus or the pathogens and the infectious diseases to come to sort of a compromise. Because if you take a look at it, we talked about the microbiome, we're filled with bacteria and viruses that do nothing to us. They're part of us. We can live happily together. And the ones that we know, and the vast majority, I would say, of these agents are out there and we don't know, because if you don't cause disease, we won't analyze you. And so that's the interest. And they're also essential parts of our lives that without them, we would not function. We talk about the microbiome. There's a virome, a parasitome, a fungome, so fungi, etc. So that's, And this is very interesting, and interesting times with all our you talked about sequencing, with sequencing technologies. That is, we learn now when we can sequence genomes so easily these days. If you saw that also with SARS coronavirus, how fast we've got sequenced and how can we do it. We're going to find more and more, and we're getting better and better at seeing what are the factors that control these type of things, these diseases.
Well, very good. Guido, what's the best way for people to find out more about your work?
I would say read the scientific literature. Check check out some things like, you have the HIV/AIDS Society, so American Society for HIV Research, the CDC. They keep us always up to date of the latest and greatest developments in those areas. In Canada, we have the Canadian Foundation of AIDS Research, CANFAR. And, we talk about HIV and AIDS, and we talk a lot about the biology, but there's also a lot of social issues. We talk about social determinants of health and who are the people who are getting infected. It's still a disease also of the disadvantaged, that people are at a higher risk in particular, or they don't get treatment. So, and the stigma around it, those are things beyond that are also very important. And luckily, some of those things have changed, but we still have a long way to go with regarding to the stigma. We have some good therapies to keep people alive, but those therapies are only, of course, available to more developed countries, and we roll them out in developing countries, but they don't have the full access to all the different drugs that we have. Cost is a reason, logistics is a reason, but those are the things. So yeah, I would just say follow some of the AIDS Foundation, so it's AMFAR, American Foundation for HIV/AIDS Research, and they are very good, and they can update you on all kinds of things with regard to social aspects and the therapy and the latest development. And so there are also, take a look at some of the scientific literature, some of the science literature, you've probably browsed through it quite a lot. Some of it is very hard to read, but there are a lot of things that in these scientific journals that are really, I think, easily comprehensible by anybody. And it's not because we're so much smarter or dumber. No way. Just keep yourself informed. But that's sort of where we can sort of take a look. You can check out on the web, on the subject areas that I discussed, and you probably will find me as well as other. I'm just one of the many people who work on this, and I wouldn't say that I'm the guru on everything, but I think these are some of the things that you can find. I wouldn't say that this is group work.
All right, Guido. Well, thank you for coming on the podcast. I appreciate it.
Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.